Oh, I was muted. So after some AV challenges, let's get started. Welcome to your Healthspan seminar that is hosted by the UCI Center for Healthspan Sciences and the UCI Division of Geriatrics and Gerontology. I'm Mahtab Jafari. Before this recording started, I had the pleasure to talking to some of you. I'm the director of the center, and I'm also a professor of pharmaceutical sciences at the School of Pharmacy and Pharmaceutical Sciences. And I know firsthand that we are an over-medicated nation, and it's good to sometimes focus on wellness, or at all times focus on wellness, because as we all know, our healthcare system really focuses on treating a disease, not necessarily preventing it or early diagnosis. So here at UCI, my center, our Samueli Institute, and many other units on campus are trying to change that paradigm and hopefully become a more wellness-oriented community. The mission of our center is to inspire and educate our community leaders, our community members and leaders to learn about latest evidence-based modalities to improve health span. And we bring experts to our health span seminars. I should call it your health span seminar. So we try to bring experts from various fields such as medicine, psychology, AI in healthcare, health span pharmacology, and health coaching. And we collaborate on a number of research and educational programs to offer these educational activities. And we also offer courses. Some of you might have heard about Life 101. It's a course, it's a self-care course, mental and physical self-care course that we offer to our undergraduate students. And the course is offered to UCI, not only UCI students, but to students from all 10 UC campuses, with an average enrollment of about 800 students. The course is also offered for free on Coursera. And I couldn't help but check this morning to see what is the total enrollment, and it is very close to be 20,000. So we really try to reach a very wide population and also a large population. The center also developed a course titled Healthspan Sciences. The course was offered to our students once, and stay tuned for more news about this course because we are expanding it, and we are hoping to offer it to larger crowds. So without further ado, I am going to introduce our speaker for today, Dr. Nathan Price, who is a professor and co-director of the Center for Human Healthspan at Buck Aging Institute, or Buck Institute for Research on Aging. I still call it Buck Institute, but Buck Institute for Research on Aging. He's also the Chief Scientific Officer at Thorne, which is a wellness company that serves millions of individuals. You have seen Dr. Price's bio on the announcement for this seminar, so I am not going to cover it line by line, but he's extremely accomplished, and his accomplishments include co-authoring a book with Dr. Lee Hood titled The Age of Scientific Wellness, and he's also recognized as a leader in health and medicine by national academies. So I can go on and on, but I'm going to just ask you to read the bio again. But what makes him an exceptional speaker for your Healthspan seminars is the work that he does, and he bridges AI and healthcare and system biology and personalized health strategies. And what I like the most about his work, which is on the cover of his book, that everything is in your hand, meaning that hopefully these modalities become available to everyone and not just a few who can, for instance, afford practicing scientific wellness. So Dr. Price's presentation will be 30 to 40 minutes, and we will have plenty of time for questions. We also have a number of Zoom participants. So for those of you who are joining us on Zoom, please type your questions in the QA and we try to answer them as much as we can. So let's welcome Dr. Price.
Great. Well, hello, everyone. It's wonderful to be here. Love coming down to UC Irvine and visiting with a number of friends and colleagues here. So there we go. So we're going to talk about optimizing human health span in the age of scientific wellness. As was mentioned, I do kind of wear a couple of hats, so I'll just mention a little bit about kind of where I'm coming from. So the Buck Institute, if you're not aware, is a private nonprofit research institute in Marin County. Kind of an interesting story. There was a couple, they were very interested, they had a little bit of, they built a little bit of wealth. They had about $10 million and they left it to help the aging in Marin County. Then after they died, oil was discovered on that land and all of a sudden it was worth like $350 or $400 million or some crazy number. And the foundation didn't love the notion that they were going to only support the health of very rich individuals in general that live in Marin County. And so anyway, I won't go through the whole story, but basically that led to the establishment of the world's first institute dedicated to research on aging with the goal of trying to help people with aging throughout the whole world. And so the Buck is quite a special place that I've been at now for about 6 months, so I'm pretty new there. I also have another hat, as was mentioned, as I am the Chief Scientific Officer of Thorne. So I'm very interested both in like trying to drive science, but also trying to get solutions that we can get out to millions of people over time. Okay, so I'll start with just a brief note on, what is aging? And the definition that I like the best is basically just thinking about year over year that the average, that the likelihood that you're going to die from various different diseases just ticks up, right? And you can see the, I guess the mic is right there. And so you can see kind of how the, how that ticks up. I don't know why this sticks out to me. I remember as an undergrad learning about light bulbs and how light bulbs were, will burst. Light bulbs will burst just as likely on day 1 as they will on day 30,000. In other words, there's no aging to them. It just has to do with the power grid. That filament isn't getting older or anything. And so it's very dichotomous to think about what it means to not age compared to what it means to age. And it really is, at least in my mind, related to that uptick of everything's getting more and more probable to break down. So we want to think about how we can address that. So we think about longevity, right, which is the total span that your life lasts, but we're really mostly focused on health span. And one of the things that was interesting, I was going to give a talk on scientific wellness at RPI up in New York. I was driving up, I was catching up on some reading, want to stay current. I'm slightly behind. This book is 2,000 years old. It's by one of the Stoics called the Moral Epistles. And so I'm listening to it, and just by happenstance, it goes on to epistle, I forget what, 54, whatever one it was, about longevity. I thought, oh, this is interesting. Let's see what people were thinking about 2,000 years ago. And one of the things that he said that I really liked was, for it makes a great deal of difference whether a man is lengthening his life or his death. And that's really what we're talking about here, of course, which is how do you extend health span, and rather than have that last part of life linger on. And so it's really about how do we make that healthy part of life as long as possible and that death part of life as short as possible. So obviously not a new concept, a concept that's been around a long time, but do we have tools now where we can start to attack that in new ways? And so the way that I think about this is something that we call scientific wellness. Mahtab alluded to this. Obviously, a lot of people at UCI are very focused on these kind of ideas as well, which is to move away from our disease care system and towards something that's really focused on wellness or health. Right before the pandemic, I was on a panel with the emeritus chairman of Harvard Medical School, and he put this in a way that I really liked, which is that healthcare is the only industry that does not study its own gold standard, which is wellness. And so if you think about that from a data perspective, we had an incredible journey in medicine through the 20th century. So if you go back into early 20th century, this is kind of a morbid statistic, but every third coffin would have been filled with the body of a child who had died from an infectious disease. So if you think about that now, 1/3 of all of our children would be dead right now, and they're not, unless this is an incredibly unlucky room, which I hope it's not. That was an insanely great achievement of modern medicine, attacking infectious disease and eliminating the ways that we really were dying. And if you look at the top 5 diseases that we were all dying from in the early 1900s, we don't think about them very much anymore. Now, that said, we've taken that kind of find the pathogen and kill it and turned that into find the target, drug it, and applied that to chronic disease. And we have had nothing like the success in chronic disease that we had in infectious disease. Cancers have not been eliminated. Alzheimer's has not been eliminated. Diabetes, and many of these things, as we know, have gotten worse. There's many, many reasons for that I won't go into at the moment, that a lot of us will be aware of. So one of the things that in this book that Lee Hood and I wrote together called The Age of Scientific Wellness that we argue for is that if we're going to attack chronic diseases, that we should really be doing a very different style of paradigm or approach to how we do biomedicine. And that starts all the way from the research level through how healthcare is delivered, meaning that on the left here, if we think about a medicine aimed at these chronic diseases, or what we'll call here 21st century medicine, we really want to be about the maintaining of wellness, understanding what's happening in a very personalized way, building systems that are predictive, that let us do prevention, and trying to do that in a precise way, leveraging AI, big data tools, lots of things that we have at our disposal now that we didn't have before. And so let's dive into that a little bit. So one of the things that I'll say is that we are starting to see a shift more broadly in this space towards wellness. This has happened quite a bit over the last 10 years. This on the left is pretty interesting, I would say. This is a forecast from a big consulting firm called Deloitte. It does a lot of industry consulting. And one of the things that they're forecasting is they look first currently, and this is as of 2021. So we spend about $4 trillion on healthcare in the US, as we probably all know. About 2.3 trillion of that is on care of illness after you're significantly sick. Delaying symptoms right after you've already had them starting and trying to do that is another 765 billion. So you've got 3 trillion basically there. And then at the top, you have a little bit on restoring health and promoting health. And what they're predicting is that you're going to get a shift to about $2 trillion towards preventing health over the next 2 decades. Now, you can either believe that or not, but one of the things that is nice is that you're starting to see at least the inklings of this, and you're starting to see more and more places like the Samueli Institute, like Mahtab's Institute here, like all these things that are happening, and we're starting to see that shift. On the right is an article that I wrote for Scientific American around the same time, which goes through about 50 different startup companies that have emerged in this like wellness or scientific wellness industry, so that you can have devices that are measuring, we know all these kind of wearables, AI systems that can give you lots of access to incredible amounts of information that you didn't have before, et cetera, et cetera. One of the good examples of this, I think, just to make it more concrete, is Alzheimer's disease. So I think it's incredibly illustrative of the difference between late stage and early stage thinking. So if you think, and this is a piece that I wrote with Lee for the for LA Times last year, but basically if you go through this, if you look at the amount of money that we have spent collectively on clinical trials over the last 30 years or so, you're talking hundreds of billions of dollars, massive effort, most of it aimed at amyloid, late stage care. We've gotten almost nothing out of that in terms of benefit to patients. A couple of drugs approved last year or a couple of years ago now. Modest improvements with side effects, very costly, right? $27,000 a year, something in that kind of a range. Conversely, if you look at things like prevention, so the finger trial is the most successful trial, I would argue, ever in Alzheimer's. You get years of benefit, and you're talking about diet, lifestyle, exercise, some supplements, maybe like just a very small kinds of things that were happening. And the reasons for this are pretty clear, but worth thinking about, which is when you're trying to treat Alzheimer's, you have a bunch of neurons that have died. Your synapses, a bunch of synapses are gone. Once that has happened, nobody knows how to put those back together. And the notion that you could take a small molecule drug and like recreate all the synapses that you lost and the patterns that encoded your life and memories and behavior, I don't even know how, I don't even know how that would happen. I could imagine some sci-fi ways we might think about it. You have a mapping of your brain from before, nanobots with like, like stem cells and stuff that you could like deploy to like regrow or like, but you probably need some sort of pretty, very complicated solution. Prevention, on the other hand, is keeping your neurons from dying. You don't need any information about all that encoding. You just have to stop that process. That's a way, way easier thing to do than is treatment. And that's why when genetics was really getting bandied about in the early days, and there were all these news reports and they'd say, don't go get your genetics because you're going to find out you have risk for Alzheimer's and there's nothing you can do about it. And that is actually completely false. There's no drug that you can take where you won't get Alzheimer's, but there's many steps that you can take that will lower the risk, push it off into the future, and so forth. And we've gotten into that in quite a bit of detail. Now, that dichotomy is not something new, right? Something we've understood for a long time. That's basically the story of Humpty Dumpty, right? It's hard to put the egg back together, easy to keep the egg from breaking. Pretty much a lot of that is the same with the brain. And so as we focus on prevention, you can do all kinds of things to keep that process from going on. And we could talk more about that. Okay, so coming back to like the broad topic of scientific wellness, as I mentioned, we want to be focusing a lot earlier. So one of the things that you've got to do is you've got to generate a lot of data throughout the course of a person's life and not just late stage disease. So if you think about the way that we spend money primarily at the NIH and so forth, we primarily focus on late stage disease. Reasons for that are kind of obvious to people in research. You're going to get your 1 R01, you're putting in your grant. You need a big difference, so you kind of find case controls. A study that says, I'm going to monitor people for 20 years and watch them get into Alzheimer's requires a very large population, a long study. You're now saying, instead of writing your grant, you're saying, hey, I need $500 million. They say no. And so you basically end up with kind of a desert. It's getting a little bit better now, but for a long time, pretty much a desert. And so what Lee Hood and I were really focused on at this time was trying to think, how could we get a lot of data from people as they were just going about living their lives that would help to build forward towards what we were calling scientific wellness. And so we launched a project called the Pioneer 100, where we captured data from about 100 people and started gathering it. And then we started a company, no longer in business now, but called Arabelle, where we were able to take about 5,000 people through a program like this over the course of 4 years. And I'm going to talk a lot about some of the things that we learned by analyzing those data. So the kind of things we were looking at, this was whole genome sequencing. So you get millions of differences between people at the level of SNPs. Labs, about 1,200 different analytes out of the blood. We were taking these in the early part every 3 months, and the later parts of this every 6 months, doing gut microbiomes as well. And then continual tracking with Fitbits and other types of wearables. And then the idea was, if we could provide value to people for going through a scientific wellness program or making a difference in their lives today, that maybe they would stay with us over the course of time. And by doing so, you create what we call personal, dense, dynamic data clouds. Personal because it's about the individual, dense because there's a lot of stacks of information. And in research, that's one of the things I'm a huge believer in. And I'll show a lot of examples of that here in a moment, which is how much you can learn by having deep data, a lot of this different kinds of measures on the same people, and then dynamic in the sense that we're watching it over time. So then this really becomes all about now taking these types of data and applying computational approaches and AI systems and so forth to try to understand as much as we can about those trajectories of health. Okay. So that's really the setup to now starting to go through a number of different things that we've learned by analyzing data on thousands of people throughout a bit of their life. And I'll start with on microbiome and metabolism. So one thing we learned that was quite interesting is we developed a score for a microbiome. It's a little bit of a weird metric, honestly, a little bit of an unusual one in the sense that we called it microbiome uniqueness. So it's really how different is your microbiome from anybody else's in the cohort. And one of the things that we saw was that starting out at around age 50 or so, that this starts to tick up in life. And so the older you get, then the more that this goes up. It turns out that that's only true if you stay, as we would call, reasonably healthy in this trajectory. It goes away if you're on more than 5 pharmaceuticals. That cohort doesn't show this. Folks that are hospitalized, those don't show it. And I'll show that here in a minute. But otherwise, we see this, this tick up. And so when we looked at this then in a validation cohort, we then evaluated in the elderly this microbiome uniqueness score. And this is what we saw, which is that if people, if their walk speed was high, they had high microbiome uniqueness. If they rated their health as excellent, if they were overall composite healthy and so forth. And if we looked at just the diversity of the microbiome, which is another measure, there was no signal actually. So this uniqueness score, which refers a lot to this diversity of low abundance microbes that start to take over more and more of the microbiome as you get older. And that signal was strong enough that when we applied it into the Mr. Oss cohort, which is a cohort of over age 80 of, in this case, men suffering from osteoporosis and sarcopenia. And when we apply it to that, what we found was that it was predictive of all-cause survival, if they would, who would go on to live for the next 4 years. So pretty interesting. And what it kind of told us was that the microbiome was, there's at least a lot of interesting signal in that as you're looking through aging. And that was supported by a Catalyst Award from the National Academy of Medicine. So one of the things that was interesting was that even though the species of the microbiome started looking different, right? Your microbiome was looking a lot different than other people's. If you looked at the metabolic transformations it was doing, those were largely conserved. And so there's a bunch of metabolites that are made in the microbiome that come into your bloodstream that are either positive or negative for your health. And so some of those are, if you're making like indoles or trying to understand. Anyway, TMAO here is supposed to be red because it's bad. I have no idea how that changed. Weird. Anyway, AI at work. So we'll argue about that. So if you're looking at this, so you'll have things that are negative to health. TMAO is actually a really interesting example. So if you're eating, say, red meat that has carnitine in it, if you have certain kind of bacteria, some of you will convert that into trimethylamine, which your liver then oxidizes. This is a risk factor for cardiovascular health. Another interesting one is that phosphatidylcholine, which is one of the compounds that has some evidence for being protective, of one of the things you can do for trying to stave off dementia, not on its own, but one of the multi-factors. What's interesting is you could be like taking a supplement or trying to eat more eggs or something like this to get the choline benefit for your brain. But if you have certain kind of bacteria, they will actually eat that compound and they will turn it into, again, trimethylamine. You end up with TMAO, you're poisoning your heart. And so there's all kinds of things like this on personalization that make a big difference in terms of, if you're trying to aim at a target and missing it. So this relationship between the microbiome and the metabolome is very strong. And so one of the things that we saw, and this is published in Nature Biotechnology in 2019. On the other one I should have mentioned, if you want the details, was Nature Metabolism in 2021. But in this one, it turned out that you can predict half of the variance of the diversity of your microbiome, which is associated with your health, but you can predict half of it off of just looking at metabolites in the blood. And in fact, I'm actually a pretty big believer that one of the best screens for the health of your microbiome is actually a blood test where you're looking at the compounds that are being made in the microbiome, whether positively or negatively. And so when we looked at this, so both the prediction of the microbiome diversity as well as it being predicted from the metabolome, what we saw was that was robust under all kinds of different conditions. So if you had abdominal pain, if you're on antibiotics, which really decimates it, if you're constipated, diarrhea, anyway, and so forth. And so it was very robust. In fact, we only saw one condition in which the relationship was totally obliterated. And that was in extreme obesity. So if you had a BMI higher than 35, what the World Health Organization defines as type 2 or 3 obesity, what we saw there was that metabolites that were really predictive of microbiome diversity the signal there went to basically 0. And we think this is because of maybe high inflammation states that are triggered at that level. And so it became interesting then to look at whether or not we could predict at baseline for these thousands of people coming through these health programs who would go on to lose weight. And we tried a number of different things. So we normalized for BMI, right? Because body mass index or just how overweight you are is itself a strong predictor. But once we controlled for that, we then said, is there a signal in the metabolites that related to whether or not people would go on to lose weight? And the answer, very surprisingly to me, was no. At least we weren't powered at a level to see that. There's a whole digression there in terms of, well, I won't go there. Well, I'll just say one thing, because in genomics, what we've seen is that sometimes you see no signal, and then when you get millions of people and you can apply like really deep learning to it, all of a sudden there's an amazing signal, but it's very complicated. So there might be some signal in the metabolites, but it may be too complicated to see yet. But anyway, it showed no predictive ability. All the proteins, the hundreds of proteins we were measuring, no predictive ability. Even when we asked people just to report on diet frequency, what they ate, that again was not predictive. It was trending a little bit, but not predictive. So of all the data that we generated, the only thing that predicted at baseline whether or not someone would go on to lose weight as they were going through the wellness program was the microbiome. And so that was, and it's particularly the gene content. And there were two things that really mattered. One was how fast the microbiome was growing, particularly something called the Bacteroides. And then the number one factor was if you look at the gene content of the microbiome, if you had a microbiome that was more likely to break down complex carbohydrates, right? So you eat a sweet potato. For some people, it will break down that microbiome. It will break down that complex carbohydrates more into simple sugars. And other people will break it more into short chain fatty acids. So if you go into simple sugars, it's much harder to lose weight. It's going to cause insulin spike. It's going to drive more hunger pangs. You're going to have a cycle that's tougher to fight. If it makes short chain fatty acids, then that is not happening. And what wasn't known at the time of the paper, or at least wasn't known to us, was that butyrate, which is the most prominent of these short chain fatty acids, one of the things it does is stimulate the secretion of GLP-1 in the bloodstream. And so that is, of course, all the rage these days. But basically, that means that for the same action, the same thing that you're doing, you'll have a quite different response based on your microbiome. And so that's another really important aspect of personalization. And there are ways that you could try to shift to have a microbiome that's more favorable rather than less favorable. Akkermansia, for example, which is sold as a probiotic, not by me to be clear, for example, is one of the species that is enriched in the people that got breakdown into short chain fatty acids and so forth. So another example from the microbiome is that the gut microbiome is strongly, let me back up. Let's look at people that take statins, right? So statins, millions of people are on statins, of course, to lower LDL cholesterol. And so what we did here was just cluster these individuals into 4 broad groups in terms of the kind of microbiome that they had. And what we saw, which really stood out to us in a big way, was that for one of these groups, you get double the lowering of LDL cholesterol on a statin that the other groups got, okay? And this is observational data, to be clear here. But when we looked at that, we saw a very big signal here. Now, the other thing that happens with statins, there's a lot of benefits to taking a statin, which is why they're given, but there are some negatives. And one of those is that you see a 9% increase in the rate of diabetes on people with statins. And so again, we look to see, was there differences in those across these microbiome categories in diabetes markers? Because again, we have this really dense data set, so we can analyze this kind of stuff really quickly. And so what we see here then is for 2 of these groups, there was a significant significant uptick in diabetes markers, insulin resistance. But for the other two microbiome groups, there was no observed effect. So this is still very early. We're actually putting in a grant proposal to do a follow-up clinical trial on this right now. In fact, we have a meeting about that here at UCI later this afternoon. And so, so this is all very interesting. And one of the things I want to point out about the niceness of having this kind of dense data, this kind of observation that we see here is not as good as having run a randomized clinical trial of giving statins and so forth. But if I want to run this as a clinical trial, I have my hypothesis. I have to write up the proposal. That's going to take 6 months. Then I'm going to submit it. Then it's going to get rejected. Then I'm going to submit it again. Then it's going to get hopefully accepted. Then I'm going to start the trial. Then I'm going to recruit. Then I'm going to pull people. And, it's about 7 years maybe for me to get the answer. This took me 2 weeks to get the basic answer. It took longer than that to write the paper and everything. But we literally, because you already have so much dense data, you can ask hypothesis, likely answer really rapidly, because we didn't have to go get people, because we already had the microbiome, we already had the usage of statins, we already had the diabetes markers, we already had. And so as you start building out these kind of dense data sets, you can use them to answer thousands of questions and get like insight before you then go launch, your clinical trial. Okay. All right. So shifting away from the microbiome now, let's look at something like biological age. So biological age is a concept of, what's the age that your body says that you are as opposed to the age that the calendar says that you are. There's all kinds of ways to do this. Some of them are kind of gimmicky. Some of them are kind of interesting. But it's basically an analysis of what changes in your body over time, right? If done properly, you can use this as a metric to track over time, and I'll explain what I mean by properly here in a moment. And you can base this on lots of different measures. And what's important to know is that if you're basing on different measures, you will get different answers. There's no like universal answer to this right now. And so the way that we ended up doing it was based on clinical lab measures, at least the one that we did at Thorne, mostly because the clinical lab measure one is the most related to health outcomes today. But the way that we do this, and the reason I'll say from a proper perspective, so here in this paper, we published this in the Journals of Gerontology in 2019. So you just get your big cloud of data. In this case, we used all the data points we had. So there was, so we're looking at, that 1,200 analytes in the blood. And so you just fit, you fit a curve to it. So you're just saying like, how old would I predict you were if all I knew about you was this cloud of data? And you can always do this, right? And so, so it's not meaningful at this stage. But one of the other things that we did is we used something called the Clamara-Duval algorithm. And I just want to point out one thing, which is that it forces this, it forces the slope of this line to be as close to 1 as possible. And so what that means is that for a group of people, we would expect on average this to tick up at 1 year per year. Okay, and we enforce that algorithmically in terms of how we learn that. And that becomes important. So then what you're wondering is that meaningful? If I'm higher or lower, is that telling me anything that I care about? So we did two tests of this basically. So the first was, because again, we had all this really dense data, we looked at incidence of disease and asked, are people who have disease versus not in these cohorts, do they have higher or lower biological age? And what we found was that in every single case where there was a statistically significant difference, right, some no differences observed, but every star that you'll see on this list, every time it was statistically significant, it was associated with higher biological age. And we saw no example of that going in the opposite direction. So that was kind of a sanity check to say, okay, it seems like, okay, so a higher biological age was associated with negative things like disease. More interestingly for me was this modifiable? So people were going through a health program, our health program at the time, so we were hoping it was making a difference for them. But then we looked to see what happened to this biological age metric as they did so. And this is what came out. So what we saw was that on average, the biological age of that population went down by negative 0.16 years per year. So compared to expectation, that's a 1.16 year on average benefit or lowering of this biological age score. Women did particularly well. The score lowered at 1.5 years per year. Men didn't do quite as well, but they were going up only 0.2 years per year, as opposed to 1. And so this was like a measure of health. By the way, this is of course not something that you can continue indefinitely. You know, the more you're below your biological age, the harder it is to move. We did a lot of analysis on that, like looking at regression to the mean effects and so forth. One interesting outlier here, though, at least to myself, was that my biological age went down 2.25 years per year for 4 years in a row. So it went down by 10 years during the period that I was one of those data points, which I was very happy to see and surprised. And this was a metric that we didn't have at the time, so it wasn't coached to specifically. I'm going to skip this in the interest of time. Okay, so that's a little on biological age. So now I just want to talk a little bit about genetics and looking early. So one of the elements that's really interesting is that genetics has a big effect on all kinds of different blood measures. So let's look at LDL cholesterol again, just because it's a very common example. And so you can develop a polygenic risk score. So if I don't know anything about you, your lifestyle, what you eat, nothing except your genome, at the time, we could predict about 12% of the variance in that measurement, which was enough to take you from below clinical range to above clinical range. So it was a meaningful amount. By the way, the new polygenic risk scores now, we can capture about double that amount now. So it's become more and more predictable. But you can see here that then if we look at the actual blood level, we saw that people in the lower polygenic risk scores had lower LDL cholesterol and on up, as you might expect. So then the interesting question is it different, or do people respond differently with the same LDL cholesterol if they have a different genetic risk number? That's really important because we measure LDL cholesterol in millions of people and prescribe drugs, and we don't measure the genetic score. So is that important? And so what we found was that the difference between your genetic score, your genetic prediction, and your actual value was highly predictive of whether or not you could lower LDL cholesterol by lifestyle. So what we saw was that people in this lower quadrant who had elevated cholesterol, but their genetics predicted low, they saw significant lowering when they went through a lifestyle program. And conversely, people that had this high genetic risk, neither of these was statistically significant. So there was no statistically significant ability. That's on the top 40%. So you're talking about, again, across the population, millions and millions of people. This was a study in 3,500 people. And so when we looked at that, that was, to me, this is a really important measure because it's not only true for LDL cholesterol, it's true for HDL cholesterol. It looks like it's true for hemoglobin A1C. So you can just imagine there's an entire category of variables in medicine completely unused today, which is highly valuable for personalization, which is not only what are you at risk for, but you could take all these biomarkers of health compared to the genetic risk, rank order them from the biggest gap to the smallest, and you would get a rank ordering of all the things that were most likely to change by lifestyle intervention. What's the easiest thing that you could do to make an impact on those measures immediately? And again, that is essentially not used. Let's see. I want to make sure I have time for questions, so maybe I'll skip this part. And then just say, I'll do this like incredibly briefly. One of the things that turned out to be interesting from this cohort is that rather than say, I'm going to wait and watch for disease to develop, I can just use a genetic risk profile to say, these people are much more likely to get Alzheimer's and these other people are much less likely. And I can start to look, even in asymptomatic people, about what are differences between populations that are at differential risk, right? Just correlate that with genetic risk scores. So then we could say, all right, well, let's find that right away. So we did that for 54 different traits and conditions, and we ended up finding about 750 markers that were associated in the blood with the early, which as candidates for really early stage markers because they're related to that genetic risk. These turn out to be quite interesting. There's a few examples here. One, I guess in the interest of time, I'll just point out one at the bottom. There's only one protein whose level in the blood is correlated with genetic risk for coronary artery disease, and it's PCSK9, which of course turned out to be the biggest blockbuster drug target in that space over the last year. There's a bunch of interesting stories in here. This is all published in PNAS in 2020. So if you want to see the details on that. And then here, just one simple example, we could also just monitor for early warning signs. So as we had someone coming through the program, so here's a woman age 57. We find she has very elevated, in this case, CEACAM5. This was a retrospective analysis of getting proteomics. So we weren't actually giving this back to people at the time, just to be clear. We weren't allowed to legally anyway. So here, but anyway, just to give you a sense for what this looks like. So here's a woman, time point 1, time point 2, time point 3, you see that elevation. Time point 4 stays elevated. She then is diagnosed with pancreatic cancer and dies, which was very sad. But basically, that marker, CEACAM5, which is a well-known marker of cancer metastasis, was elevated in her for 2 years. And she had no symptoms. And by all of her classic clinical measures, she was very good. She was in very good shape. She was one of the healthiest people in the whole cohort by any other measure, except she didn't know that that was ticking along. Another one, man, 69. Again, here elevated. And by the way, I say that that's elevated 2 years. That's just the farthest back we had. Could be 3, 4, 5. I don't know. I don't know how much earlier it was. We didn't catch the transition. We just know she already likely had it. Same thing here, a man, CEACAM5 up here, time point 1, 2, and 3. Then it jumps a little higher in 4. Again, that represents about a 2-year period, diagnosed with lung cancer, so metastatic cancer. Then another woman, 57. She starts out really low and then jumps. So this is probably when her tumor starts going. The important thing there is that she's still in the normal range. But just that jump itself is very unusual. We did a statistical analysis. How likely is it to see a jump that big within the normal population? And it's very unlikely. So you can build, differential predictors on this. Then you can see time point 3. So hers is just going incredibly aggressively. And that one, we didn't have quite a 2-year lead window, at least with this marker. And the interesting thing about CEACAM5 is it's a known metastasis marker. But what wasn't appreciated was that how much earlier it appears that it pops up in this process than was previously known. Now that's only 3 examples. So we only had 3 people go to metastatic cancer. This happened in all of them. And we measured it in all the other people. And we just didn't see it elevated in any of the others. So preliminary, but to me, a pretty given that we already know CEACAM5 is such a good marker for metastasis anyway. If my CEACAM5 jumped high, I would worry about it. Okay, so with that, I'll sum up. So we went through a number of examples, sort of what can we learn by looking at data longitudinally on a lot of people, how we can use genetics, dynamic measures to get clues towards early cures, the importance of the microbiome. And I'll just say one brief word about where the lab is heading, is that we're really leaning very deeply into this new AI space. And so developing tools that are going to let us go incredibly much deeper into analysis of this type than what we could do from like a statistical or a data machine learning approach. And I'm happy to talk about that in question time if people want to. Thank you. We're just sitting here.
Thank you so much, Nathan, for your wonderful presentation. And I'm going to go on the Zoom to see if we have any questions. So for Zoom participants, please feel free to type in your questions in the QA, and I see 2. But we are going to acknowledge our colleagues and friends who joined in person first. So do you have any questions? You see, you get a priority with QA. Then this is for Zoom participants. Any questions? Yes. Yes.
Can you elaborate on the digital twin capabilities that are forthcoming and may help us?
Sure. During the period, when I was at Thorne, we were working together with a startup company. So I spent about 3 years building out a digital twin for Alzheimer's and brain health. And that ended up being, I think, really ended up learning a lot from doing this. So one of the things that, so I'll get into a couple of things on that. So one is that you can make predictions from molecular perturbations through like the number of years of benefit that you might expect on a per person basis. So we actually developed with this company a capability that would let us like forecast, like if you do, these sets of things, whether, how much it would make a difference. So I'll just give some examples. So by looking at the digital twins, one of the super interesting things that came out is that we were focused very much on like lifestyle and because I was at Thorne, like supplements and things like that, like kind of everything that we could do in the wellness space. And what we saw was that almost all single interventions would have like very minuscule effects. And so, but when you started doing targeted combinations, at least in the digital twin simulations, you would start to see an effect. Like you could take 3 things that had basically no effect individually, and all of a sudden it would have 6 years benefit in 4 individuals. And we compared this against a bunch of population data. So so that's one thing. A second is that it was a really incredible experience, at least for me, in terms of like changing the way that I think about that disease. So the way that we built this capability was a lot of like ordinary differential equation modeling of homeostatic mechanisms in the brain. And then you deal with uncertainty with a Bayesian layer network overlay and stuff like that. But I won't get into too much in the weeds on that. But what we started to learn was, one, that certain things about Alzheimer's, we could not figure out any plausible mechanism for it to be true. One of which is that amyloid is causal to Alzheimer's disease. That's, of course, starting to be more and more a contested issue. But one of the things was when you try to build a biophysical model like this, we could not figure out any mechanism by which the aggregation of these plaques could be a driving cause of Alzheimer's and make the kinetics, the dynamics, the time series make any sense. Because if that were the driver, Alzheimer's should progress much, much faster than it does, for example. Conversely, what we found was that there were other mechanisms that were highly predictive of Alzheimer's disease. The main one being that I think the central driver around Alzheimer's is really around energetics, right? Your brain, I know it's a long answer to this question, but your brain consumes 20% of your body's energy, right? It's 2% of biomass. So you have to feed energy up there all the time. But so what happens is that as you get older, and this is all measured, the amount of oxygen that you can perfuse into your brain goes down. Your arteries start getting a little bit more clogged. Their penetration into certain parts of the brain don't. And so this is like the primary trigger for those neurons dying. In fact, there was a study that came out from UCLA as we were building the twin models that showed imaging data from 8 years in advance of Alzheimer's. And they showed these pockets of what they called hypometabolism, these low metabolism areas where the cells would die, and that was like the trigger. That matched exactly with what we were finding. Conversely, it now explains why ApoE matters to Alzheimer's disease. Because what happens is that when you put together these equations, you start putting together the model around solving that energy balance. It turns out, so you have neurons and you have like about 9 astrocytes that support it from an energetic perspective. And when we ran the simulations, it turned out that to keep energy generation in neurons efficient under low oxygen conditions, you want to keep cholesterol levels in astrocytes low. Well, ApoE has a role in the transport of cholesterol. ApoE4 is super slow, and so that cholesterol stays high, and the efficiency of energy production in neurons is thus not good, and a higher fraction of them die. And we can simulate this whole thing. And then, but ApoE2, which protects you, it does it really fast. And so that keeps that energy generation done. And if all you do is take those 2 facts on a backdrop of 10 million digital twin simulations, it recapitulates the Kaplan-Meier curves of the age of onset of Alzheimer's across all the known genotypes. And it does it for ApoE2 2s, 2 3s, 3 4s, 4 4s. And it also works for early onset Alzheimer's. So we put that in. Anyway, I could give a whole talk on this. The only reason I don't is because, anyway, the end of the contract with the company and us was a little bit messy. And so it's hard for me to progress. I have to rebuild all this stuff. And so I have to do it in a different way. And so
Nathan,
I used to
just
talk
so
about
we
it all the
can-
time, so it's actually a sore spot for me, which is why. I really like
I was
buildings.
gonna say that, I'm just inviting him right now in front of all of you to come back and just talk about
I
that.
just talking about that. It's actually a great talk. It's a really, we learned so much by doing it. It was really fascinating. So there's a lot that's building out on the digital plans.
So I'm going to the questions from our Zoom participants. There are many of them, so I can't just ignore them. The first questions I said I will answer them live, and the questions are about the availability of the recording. So the recording of today's seminar will be on the center website. I've already received some emails that the videos are missing. We are working on the website, so hopefully everything will be up and running within the next 2 weeks, including the recording of the presentation today. So one of the questions is about, and actually that was one of my questions too. You talked about the CCAM5 as a biomarker for pancreatic cancer, breast cancer, and lung cancer. The question is, if you're suggesting that CCAM should be broadly used for cancer screening, the problem is that when analyzing large numbers of cancer patients with cancer, there is far too much overlap in specificity between normals and cancers. No studies have looked at CCAM velocity and cancer risk.
Yeah, so I think that's a really good point. So this is a well-known problem in biomarker discovery, which is very true. Like you can have something that looks like when you do a pair, a pair example, you get something that looks pretty good. As soon as you say, I'm going to use this for screening, then the fact that you have way more people that don't have cancer than have cancer. And so even if you only have like a 1% error rate, you apply the 1% on the 1,000 people, you have like, and then it's, and it's 99% good on the small set of people that have it. And so your screening kind of dies. So that's true. It's a really good question. And so, yeah, I do mean to point out like how much earlier this happens, because I think that's an interesting fact. That is not to the questioners, the very good question, an argument that, hey, that's sufficient. So the point that, and I didn't get into it in this talk, I do it sometimes in other talks, is I actually think that screening with like really small numbers of molecules is pretty much, it's really hard and it's mostly destined to fail for the reason that was well given in the question. That said, what I'm a big believer in is opening up so that we learn a lot more from lots of markers at the same time. And the reason I'm really excited about this, and I didn't mention this either, but so we just got a $52 million grant from ARPA-H to help develop some of these technologies, which I'm really excited about. So one of the things that was on that last slide I mentioned is something we call the N-of-1 analyzer. So what this is going to do is it's going to take AI technologies and apply it on an N-of-1 basis into an individual. And what it's going to do is not only deploy statistical measures, it's going to employ a whole bunch of knowledge and expertise at scale to try to say in as much detail as possible what's happening in that patient. And so the reason that I'm excited about this is basically everything that I showed you today is based on like some sort of statistical analysis, okay? But statistical analysis treats every data point as if it's just a number, right? It's just a number. It doesn't care like if it's CCAM, if it's CCAM5 or it's some other protein or it's just numbers. And so what I'm really excited about now is, especially with these AI systems, we can start to build what we call in the book deep reasoning systems that in fact is empowered with an incredible amount of knowledge about the function of the proteins, where it comes up, why it's done, where is it like. So you can start to apply basically a level of expertise coupled with an infinitely good memory about all of the biomedical literature and apply that in different ways. So I'm actually incredibly excited about what I see as a whole new class of biomedical analysis. This, like, I was going on my postdoc this last week showed me one graphic slide and I went on for like an hour on it, so I apologize for that. But it's just, I feel like the kind of analyses that we're going to be able to do over the next year or two will make kind of everything we've done previously in these statistical analyses seem incredibly shallow. I was calling all of our own work shallow, and I was like, I want to preface that with, I'm proud of the work we did and so forth. But I just think that, the kind of approach that we can take now, because of like these large language models and the generative AIs and building out like expert systems that will shouldn't use that, a system that represents, experts in like a much deeper way than the old technologies. I think that's gonna let us, get at these problems in a kind of depth where we can get to the kind of specificity that we don't get in these single markers. And we're gonna have to develop all new approaches to validity, statistical scoring. Like, there's a huge amount of like blue sky, I think, right now around this area. So I'm incredibly excited about it, but there's a ton we have to figure out.
Any other questions? Because we have a lot of questions from our audience who are participating on Zoom, and I'm going to combine a few of them. Yes, please.
Talking about the AI, and
Yeah.
as a professional, there's a lot of worries about liability. And you're talking about, data validity, those kind of components. What kinds of backend infrastructure is being developed now to assume liability or at least responsibility for the data that is both input, exported, and then implemented into these solutions in the medical field and in the research field?
Yeah, it's a great question. And I don't think there are really good answers to it yet, at least that I know of. Maybe someone knows better, but I think a lot of the legal structure is still getting set up. So at the moment, we're really doing those under pure research conditions, right? So we're not, the kind of stuff I was just talking about is very much discovery. Out there in the world, there is a lot that's being done in terms of, the deployment of lots of knowledge and information, right? So, the large language models are really getting incorporated in lots of ways. There's liability issues that are related to, making sure that you keep these things under legal compliance. We are doing some, we have to be mindful of that, like everyone else, for anything that goes out there. The hallucination problem. There's also certain things that are more true statements that the AI can make that are not permissible under different laws. That drives me a little nuts every once in a while, but it's like, so I think it's very much an open question. One of the things I think is really important is trying to sort out what's really important for protecting human health and what's really being pushed because it protects certain entrenched current financial interests that are at detriment to the public. And I think there's going to be a lot of activity over the next several years trying to sort out, the rights that people have to being able to get access to deep information that they want to have access to. Anyway.
So I am going to combine like a bunch of questions here, and then I'm going to add my own question at the end of that as a summary. So when your book came out, I read it and I really enjoyed it. And then you accepted the invitation to come to UCI. So I had to read it again, this time very carefully, because I thought, okay, I better know this book really, really well. So I read it again, and I am going to summarize some of the questions here with a question that I had from the book. So we have a lot of data. We have deep data, and for those of us who can afford it, we can even get deeper and deeper. But the question that some of our audience members ask, and it's my question too, is that what can we do now? If I cannot afford, because I know that there are centers where they charge you $10,000, $20,000 to do a complete scan, genetic test, MRI, CT scans, you name it. And they can also give you a very fancy measurement of your microbiome and your metabolites. But not everybody can afford that. So if I were going to see my physician or my healthcare provider and ask them, what tests can I do right now within the parameters of my insurance, what my insurance pays, what would they be? And what are some of the practical recommendations that you have for all of us to improve our health span? I hope I did justice.
Yeah,
There are so many questions.
It's a great question. We could take it at different levels. I had slides that I moved after the last slide, you know, that went through a bunch of the like practical advice at the end. So one element is that even to this day, like, you know, all the basics really matter, right? Sleep makes a big difference. Like, you know, they did an analysis of the Stanford basketball team, right? They shoot 9.2% better from the line when they sleep more than 10 hours, 10 hours in a night. There's the really fascinating statistic that for every hour you run within a particular window, for every hour you run, you add 7 hours to your life. So that, and when I saw that initially come out, I was very skeptical, and I went and ran the numbers, and I was like, oh yeah, I guess it is. If you look at, now that works, I think, for, you know, up to you're running like 3 to 5 hours a week or something like that. I don't think it persists to like, I don't think ultramarathon runners like never die.
Right.
So that's why I said I caveat it within a window. But if you look at like the difference between being sedentary and like running 3 to 5 hours a week, and you look at the average number of years, and you do the calculation of that number of years versus the number that you employed, and you look at it, you're like, oh, okay, yeah, it's kind of that way. So you get a lot of that. So there's obviously tons that you can do for free by just optimizing lifestyle. In terms of tests, I would say, you know, some of the tests have certainly gotten a lot cheaper. You can do genetics tests now for $50 or something like that. Sometimes $25 on Black Friday or something, I think. So there are ways that you can get access to some kind of information. The AI systems, I think you have to be careful with them, but they're getting, you know, less prone to errors all the time. And if you're thoughtful and you're somewhat educated and you're going back and forth with them, it is a very cheap entree to an incredible amount of information. And one of the things that I think is really interesting is that they give us a potential to give part of the promise of science back to the public in ways that we haven't before, because the public pays for all of our research. Then it goes into journals, and then we had the problem that they were locked behind these, you know, behind paywalls. We've solved that to a reasonable degree. Right after a year, they come out. But what wasn't solved before is that they're totally opaque to them in terms of what do they say. So these kind of technologies allow you to leverage in and ask things that into that space in a way that you can understand and ask questions of. So I think those tools alone are important. For other things, you know, in terms of getting into more deep testing, certainly doing, you know, some of your basic blood work, of course, you can get covered by insurance or things of that nature. We are working to make it cheaper all the time. And then there are emerging technologies that are quite interesting. One that I was talking to Paul Yeager, who's the chair of bioengineering at University of Washington. You might know him, really good guy. But he's developed these really cool technologies of putting microfluidics onto paper. So you just put them on like a paper strip, so you can do like a droplet of blood, and you'll get these spots that will come up, and you can literally take a picture of it with your camera so that you can and then the camera will do an image processing, and you can get a concentration measure. These were developed for the developing world, so you can get these things like incredibly cheaply. So we're looking at, okay, can we do like incredibly cheap diagnostics that you could do a bunch of the times? I think the whole scientific wellness space, you know, trying to get these devices, like even the wearable devices, they cost something, but you know, they've gotten cheaper and better, and so I think there is a huge movement to try to get that down and be more and more accessible.
And this is any other questions? Because I want to ask a last question, and I apologize in advance that we are going over time because we started a little bit late due to AV
Quick question.
challenges. Yes.
Yeah.
Where are you trying to drive Thorn? Kind of right now, kind of in the future with all these, you know, third party, you know, testing solutions, AI and all, et cetera.
Yeah, so what I'm really interested in with Thorn is we're trying to pull together more for health journeys for people and try to simplify down the ability to get health testing, AI systems that interpret those health testing, and then having solutions for people. So Thorn, you know, has a very large like supplement line, for example, generally regarded as, you know, the most reputable of those companies, or at least one of the top few at the least. And so it's got a whole bunch of things that you can apply. And so basically what we think is that we can get to a lot more efficacy and precision by tying together, you know, lifestyle recommendations, but coupled with proper usage and like really tailoring that, informed by testing and deep AI systems. And what I really want to do is to try to take as much of the complexity. So I presented like today on all kinds of things that we've learned that are really important to health. But as many of the questions come up, you want to say, okay, that's great, but how can I do that how do I action on that? How do I do that? And so what I'm trying to do with Thorn is to take, more of these things and make it like super easy for people to use. And that can be through us, or it could also be through, people in the community. I'm also, very happy to try to help anyone who wants to make these things like work in the world. And I do think that both of those things are important. What I want to do is to absorb as much of the complexity inside so that people don't have to remember like, oh, phosphatidylcholine is, eaten by these 5 bacteria. Do I have, like, it's too complicated. Whereas if it's just in a program, it just happens automatically. It's already done for you. You can just know that it works.
And kind of part 2, how does kind of circadian rhythm biology alter not only your recommendations, but kind of lifestyle habits and everything you presented?
That's a really cool question. So part of this $52 million award, or up to $52 million award, ARPA is very clear about that. Not a grant, it's an award. It's up to, we have to hit a lot of deliverables. But part of that is we're doing something called the time trial. So we're going to do the most dense sampling, I think, of any population ever. We have people that are coming out of the woodwork like wanting to sign up for this trial. And I'm like, did you read how many things we're going to take? Like, how often you have to sit in that room for like 2 weeks and do nothing else because we're just going to measure you like over and over and over and over. We have like members of our board want to do it and everything. We're like, cool. So it does make a difference. We know certain things, cycle, and some of them have monthly cycles. Some of them have, and women in particular, there's a lot that relate to hormones and so forth. But even outside of that, there's the diurnal cycles of morning and night. There's seasonal cycles. And so we have that to some degree, but this time study that a colleague of mine, James Yurkovich, is going to lead out, this is going to be one of the densest multi-omic study of trying to get periodicity in all of these measures. So I'd say we kind of know about it right now, but we'll know a lot more about it in a year.
Thank you.
So the last question actually came from a few students. And I'm just going to read the question because it is from one of my former students who is now doing a fellowship at NIH. So the question is, what is one piece of advice you would give to UCI students on how they can contribute to advancing aging research, particularly by incorporating new technology? And this is actually a question that I often get, given the scope of my lab, Lifespan and Healthspan pharmacology. Our graduate students ask me these questions all the time, that if I were going to start right now to get into aging and healthspan research, what would you think are some of the topics that we should focus on?
Yeah. So there's more and more interest in aging, so there's more places that you can go and kind of get involved in people that are thinking about aging and healthspan. So you want to link up to the right kind of labs and places and, kind of get yourself into the mix, whether it's in academia or in industry. I think one of the big elements is also just considering age more deeply in your analyses. And this is one of the things that I'm really interested in. So I gave the Alzheimer's example so like genetics, we're thinking about, okay, ApoE4 gives you higher risk for genetics, but it doesn't give you higher risk in your 20s or 30s or 40s or 50s. It gives you risk later. And I explained why, at least our view of why that is, which is this lowering of oxygenation in the brain as you get older. If you have a ton of excess capacity, that difference in efficiency doesn't matter. But as you get tighter and all of a sudden it's life and death, all of a sudden it matters a lot. And I think, and this is one of the things we're trying to do in the lab right now, I think we could do a mapping across like all of genetics about not just that you have elevated risk, but when and why that when. Like what are the other things that change in your body as a function of aging that make that difference all of a sudden consequential? Because it didn't seem to matter much before. And so I think just having that as the heart of what you're doing. And so I think that's all important. And I do have one postdoc position. People are, it has certain constraints to it by NIH funding, but there is one potential position there.
This
too,
is
if
the
people wanna do
commercial
it.
break.
So if you're brilliant at computation and you like aging, give me a call.
Well, thank you so much, and thank you for staying over time. We definitely went over the time that we had allocated, but I think it was definitely worthwhile. So I would like to thank you with several thank you gifts. I'm a
Thank you.
big fan of self-care, so
Oh, me too.
I hope that they improve your health span later.
Thank you, thank you.
Thank you so much.
Thanks, everyone.